Again, INSHORE, we've been having these workshops and meetings now around the world for a few years, and we've been using my time series again to look at several different questions. So we've been looking about shapes of the range, and do they shift when they shift with climate change in geographic space? Does the shape of the range stay the same in terms of abundance? What kind of a range edge do we have? So do we have decreasing abundance across a gradient towards the range limit, or does it suddenly drop off? And this can tell us an awful lot about the mechanisms behind the range shifting species as well. So this is one of the projects that we're still working on with all of the INSHORE guys. Again, so a range of temperatures and extreme events. Extreme events don't occur too often in the UK. Like I said, the last severe winter was 1962-63, and that was a temperature event. But we've had some major storms, and they've been unprecedented in terms of wave height and wind speed in the southwest of England the last winter. And these are just two of our long-term sites, and again this demonstrates the beauty of going to the same place at the same time of year, every year, with the same people and the same methodology. This is the eminent Professor Hawkins. I like to get him on his knees as often as I can and get him working for a change. And this is at Travone in Cornwall, and this was last year, so 2013. He's doing the low shore barnacle quadrats, and he's in there, and you can see it's full of mussels. And this is exactly the same area of shore this year, and there's virtually no mussels. The storms come in and rip them off entirely. So what we're interested in now is what comes back. Will we just get a whole new mussel settlement? Life will go on as usual, or will this disturbance event change the trajectory? We might get invasives in. Often invasives need disturbance events to colonise. So this is what we're tracking as well. This involves a little bit more effort, so we're going back more than once a year to a certain number of sites, and we're seeing how seasonally the succession has changed. What we also noticed are the storm events were almost site-specific. They're all rocky shores, they're all exposed. However, we saw biological damage here. What we saw here at Crackington Haven, this is the usual way it looks. It's got a nice sandy beach here, and we just work out on this tip there. We came back this year, and the beach is gone. It's completely taken all the sand away, and there's brand new, habitat, rocky habitat for colonisation. Other places we went, totally the opposite. Where was rocky reef historically? A whole load of sediment's been dumped on it and smothered it, so there is no habitat. And again, you can see these are just the first two quadrats, so I immediately went down there and took quadrats of nothing, because that's all there was, bare habitat. And then you come back a couple of months later, the first colonising ephemeral algae have already started to move in. So this is how we're going to track it through time. What might happen with the winter storms again is it might bring all the soft sediment back in. So that habitat space might be removed. But if it isn't, we're going to track the long-term implications of this as well. And again, without time series, you couldn't put this storm event into context or work out what the damage had been. Now, I hate cold weather, and I get sick of field working in the cold weather. So I do anything to get out of the UK in winter. So when it's cold in the Northern Hemisphere, where do you go? South. So I managed to totally blag a fantastic project with NIWA in New Zealand, doing exactly the same thing. And it amazes me for a country that's about the same size as the UK, and if you turn it upside down, it's about the same shape, and it sits at about the same latitude. There's been very few studies of what lives where on the Rocky Intertidal. Now, Dave Shield and Bruce Mengie have done some work, but nobody's gone and done a marklim. Nobody's systematically just gone round and recorded what there was. Bonus. So I went the first year and thought, well, I've only got 6 weeks. Where do I start? Biogeographic breakpoints. So remember in the UK we had this break between warmer and colder waters, and that's where we see all the action going on with range shifts. I went to some of the publications from the 70s that were there, and they were based on intertidal mollusks. And this is the white lines show on the map show where the biogeographic breakpoints were in New Zealand. So I thought I'll be sensible, and I'll do survey sites either side of these breakpoints. Like Gray said, multiple sites, so you can look at within site and between site variation as well. And so I just targeted this the first year. And the white bars here on the graph show where the biogeographic boundaries were. Now what you'd expect at these boundaries is abundances would decline, depending on which side you were with your thermal affinities. We didn't see it with any species anywhere. So we didn't see it with the macroalgae, we didn't see it with any of the invertebrates. So these supposed biogeographic barriers are being completely ignored. So the question is, well, maybe these are in the wrong place, or maybe they're not appropriate in the same way that that we see all the way down the coastlines of the Atlantic. I then went and did some work just on the limpets. Now, New Zealand has 13 different species of limpets that are not found anywhere else in the world. They're endemic to the islands. And we went and did the same thing, quadrated them, counted them within the quadrats, replicate quadrats. This time, by the time I'd done 3 years surveys, I had 126 sites. So we have really, really good data coverage. I ran them through Primer to see if they cluster out in terms of islands, provinces, and everything. We saw very, very little similarity between them. So most of the populations and sites showed no trend with the geographical location at all. Really, really unusual. We never see this anywhere else. You always see either latitudinal or longitudinal clines in species distribution, nothing at all. And in the end, I checked all of these different drivers. So there were no significant patterns with individual species distributions or the total abundance of limpets or the individual species abundance. There was no relationship with biogeographical province, never seen that anywhere else. There was no relationship with latitude, there was no relationship with sea surface temperature, there was no relationship with exposure or habitat type or percentage cover of key structural and functional groups. There was no relationship with the main competing grazers, but there was some regional structure. So we were getting East Coast, West Coast was about the only thing that was coming out of this. And what this showed me is that just because we assume patterns exist and latitudinal gradients all around the entire rest of the rocky shore world, doesn't hold for New Zealand. We've got a huge amount more work to do. This is where we're at this stage. But basically, New Zealand intertidal shifts and species biodiversity and biodiversity loss of endemics is not going to be as easily to predict as we see everywhere else. They don't show the patterns we've been finding everywhere else. So we've really got to start to look now at what's causing these distributions. Policy and conservation applications of this work as well. Like I said, it's really, really important that we use this data for, to help conserve species and inform people that make decisions. We use similar techniques, so we've got new MPAs being planned for the UK. The government's been trying to cut down the number by saying there's not enough evidence. So we've given our time series to them, but they said it still wasn't enough evidence. They wanted habitat mapping. So we went out there and habitat mapped. But we also introduced some statistical rigour, so we put replicated quadrat data in there as well and time searches. So you could go back and get a little bit more information than just drawing habitat patches on maps. And this is the way you do it. So this is my map for the Isle of Wight. You go out there, you draw blobs on maps where assemblages are and where they change, and then you number them and note the species in them. And you can produce these really fancy looking maps of assemblages here. Looks great. But without the quantitative data inside that, it tells you very little. So if it's a little bit bigger or a little bit smaller, it's moved to the left, it doesn't really tell you very much. So what we're trying to do is push the government agencies slowly forward into using more rigorous approaches. This is the best set of contracts I've ever had, the Sea Caves contracts. There's absolutely nothing of interest in sea caves, hardly. It's just a bit of slime on the walls, but what a laugh. So my colleague and I get in our wetsuits, don our pink hard hats, get in tiny little boats, row in there, and we do the same thing again, but we do it in a 3D aspect. So we don't just map the 2D approach, we map up the sides of the walls and the ceilings, and again we do this, we provide them with 3D maps of where the different species are in terms of how deep the cave is, how high the cave is, and this kind of thing. Really, really important, I think, very early on in the new process to get some scientific methodology in there that people can replicate. And we also then have been doing some work for the European Union has got this massive new overarching marine policy called the Marine Strategy Framework Directive. And every country in Europe has to abide by it, and the different levels of targets for status of different ecosystems, we have to comply. So what we've used Marklin for is we've developed different indicators. So we've had an indicator of climate change based on species. So the blue dots show where it's colder water species are more prevalent, and the red dots are where we've got more warm water species. And so if this index fluctuates at specific sites, we can see where the climate change has been influencing the species present. This is just to show you that there is actually some science and maths involved in that. It's not just choosing some colours. This is biodiversity for rocky intertidal, and again we've got indicators for biodiversity. So we've got high species richness down in the southwest, And then as we go around the climate gradients, we've got much lower species diversity up north. And we've been pushing, this has just been released today, this report actually, but it's all to help government people understand how ecosystems function and how to really simply detect if they're changing and whether they're changing for the better or for the worse in terms of their policy. Now, if you think about it, in nature it's not for better or worse, it's just changing. But governments have to make decisions and allocate money and work out where to protect. And if we can say that maybe, for example, the marine protected areas, they might not be in the right location for statics to protect static species, but with such indicators, being able to track the movement means we can be a lot more informed about where the MPAs are and connectivity between them. This is where we're going to hand over to Brian and Gianluca in the morning, the biological mechanisms underpinning species responses, but I'd also do some of this as well. This is just kind of like a life cycle for some of our invertebrate species, and where climate change we think is influencing various different stages. We have biological responses here in terms of thermotolerance, which looks remarkably like the temperature graph. I think that's really neat there. We also look at how, again, coming back to this average, using means and averages, long-term averages isn't appropriate because thermal responses are not to this mean. This is the line that people use for one of these meta-analyses for the temperature that species are responding to. This is one site near me in Plymouth, and look at the temperature range over a year. How many times do these species experience this average temperature? 3 times. 3 times in the entire year. So how relevant is that average temperature to them? Probably not at all. We do all these different, again, very similar to Brian's group, measurements. So we look at population size frequencies, reproduction, phenology. So we've seen a shift in timing of reproduction during the year. It shifted early with getting warmer. Physiological performance, we've got some robo-oysters here, and behaviour as well. So we're trying to pack all of these different biological and physiological field measurements in to try and look at some cause and effect. We've seen the pattern, we've correlated with climate, now we want to see exactly whether this is the case and what biological mechanisms are driving these responses. I'm going to really briefly whiz through ocean acidification. Nobody cares about the chemistry. You know, it's getting more acidic because it's basically about all you need to know. You also need to know, never build a mesocosm. This has nearly killed me. It's been horrendous. I've never had as many problems in my entire life as I have building this mesocosm. It sucks. If any of you signed up for get out now, get out now. We've had floods, electrocutions, we've had gas bottle regulators blow up, everything's happened. But now it's working fine. And what we're doing is running long-term experiments. So this one we ran for 18 months. We had different temperatures, different pHs. And then we had the same dog whelks you get here. We numbered them all at the start. We sexed them. We put pink dots for the girls, blue dots for the boys. And then we got them to try and reproduce. We then put the next generation back into the mesocosm and measured them to have a look at what impacts there were. We did just temperature, we did just pH, and then we also crossed the treatments as well. And this is really important. The one take-home message from here, acclimation time and length of experiment. Most ocean acidification experiments that were run initially, literally ran them over a few weeks. It took our animals 4 months to acclimate to the conditions. They all stopped growing in all the tanks, even the control. But once it finally got used to the system, on they went again, and the same here with the shell width. So that's a really important, I think, consideration. You need to choose a suitable acclimation period. Real world relevance, really important for me, otherwise you've just got a nice experiment. Every time we measured the field, the animals in the lab, we went and measured the field population as well. And apart from this very start where the animals weren't growing in the lab, and you can see they still were in the field, so we knew we had an effect of the mesocosm, we got really nice agreement between what the ambient control treatment was doing and what the field population were doing. This gives us high confidence now that when we project into the future with the hot treatments and the low pH, it's probably very likely what's going to happen. This again is just showing you what we call carryover effects. These are the field capsules. So we put the field capsules, again, when they laid them in the lab, we went and got them from the field. It took the same amount of time across every treatment for the capsules, the juveniles to hatch out the capsules. Whereas the ones that were born in a different treatment, if you can see, we've got massive increases at high CO2 levels. And again, that shows us that if the adults aren't acclimated, what you get from the juvenile response is completely incorrect. We're looking at transcriptomics. This is basically a shortcut to see whether any gene groups are up or down regulating in response to the stressors. This is a cheaty shortcut for us. Looking at mechanism, you know, we could be studying reproduction, growth, whatever. We could be looking at heat shock proteins. This gives us a really nice shortcut into what mechanisms biologically we should be focusing in on, and we're still working up the data for that. We're looking at shell structure and mineralogy with Bristol University. We work with the paleo department. They usually cut up dinosaur bones, but now they're starting to look at our shells to see whether they're thicker, thinner, stronger, weaker, whether the crystalline structure has changed as well. Again, trying to look for mechanisms and how the mechanisms are manifested at the individual level. And this is all about basically making our models more real. I only put this up because it took me ages to try and conceptualize in my head where these kind of correlative models, you know, correlating climate change with species distributions versus what Gianluca will tell you, mechanistics, so starting with the individual and physiology and then projecting up. It's a horrendous minefield, but I think that all you need to concentrate on is the very bottom line, is that we've got to the stage now where we're looking at either hybrid or integrative models. So we need to use the physiology of the organisms to inform which parameters we use for correlative, or the opposite way around as well. One example of species distribution models that we've done already, and we've piled the physiology into this, and we found some really interesting results again for the dog whelks. We're looking at various different techniques with people, seascape genetics. So again, trying to see if there's any new molecular techniques that can really give us an insight into what's happening that we've never been able to have before. This is one of my final slides. It basically shows where we've been going and where Gianluca and Brian are going to carry on. So we get the weather and the GIS data. We've got all my data here, and it's going to go into these dynamic energy budget models that Gianluca's going to go into in detail tomorrow. And this just basically shows you some conceptualization of how it works. So all these parameters that are taken from the individual organism then get scaled up into a model and run with climate scenarios. A quick plug for the INSHORE network, and we've got Twitter accounts as well. I just want to leave you with a final slide, which is basically why this is the best job in the world. I took out all the beer shots. Brian said, please don't put just loads of pictures of us drinking beer, so I didn't. But apart from drinking beer, this is why studying climate change impacts on marine species really is the best job in the world. Thank you.